So good evening and welcome to the new session of Pre-Med Pro. We are continuing the chapter of botany, the principles of inheritance and variations. Okay, so today we will continue from the next part, that is the gamete formation. So let's continue. Yes, on the earlier lecture I have concluded with some basic information of this chapter, the basic terminologies. So today we will start from the gamete formation. So give the topic gamete formation. Gamete formation. Gamete formation. So what do you mean by the gamete formation? So now I am taking a homozygous gamete, that is capital T and capital T. Let it be a homozygous gamete. It is capital T and this is capital T. So 2n capital T, it is a parent. Now we come to the gamete formation here. Gamete formation. Now what happens after gamete formation? There it segregates into two daughter nuclei, daughter chromosome. So one daughter chromosome must be capital T and another daughter chromosome have to be capital T. Okay, this is a capital T gamete and that is homozygous tall, and this is also a capital T gamete, that is homozygous tall. Okay, now this one is gamete. Gamete is called as N, terminology M, gamete, and as N. Okay, now what do you mean by this? Chromosomes, here what happens? Chromosomes, write the topics. Chromosomes separate or segregate during the gamete formation. Chromosomes separate during gamete formation. Okay, in two, what happens? Each gamete carry two chromosomes. Each gamete carry two chromosomes. Okay, the number 3, chromosome public allele segregate during gamete formation, so that each gamete carry only one allele, so that each gamete carry one allele. Okay, it is the main point. Okay, now number of gamete here is 2. Number of gamete here 2. And the fifth one, types of gamete 1, and it is tall because it is homozygous tall. Okay, now come to the next part, next topic. You can say next part. Okay, inheritance of, give the topic as inheritance of one gene, study of one gene, one character, one trait. Give the topic inheritance of one gene, inheritance of one gene, that is study of one gene, one character, one gene, one character, two trait. Study of one gene, one character and two trait. Okay. Now what happens here? Let it be 2n and it is male parent. It is parent, male parent. Okay. I am drawing the chromosome. Okay, this is homozygous, that is true breeding. True breeding is happening here, true breeding. And this is 2n female parent, drawing with separate color. Okay, this is 2n. Okay, now homozygous, this is homozygous tall and this is homozygous dwarf. This is homozygous dwarf, and this one is homozygous tall. This one is homozygous tall. Okay. So I have drawn two gamete of the parental generation. One is, the left one is of homozygous tall, that is a male parent, and here on the right side is the female parent, it is homozygous dwarf. Now we do the gamete formation in first generation, filial 1 generation, F1 generation. In F1 generation, what we get? Allele from F1 generation male, okay, and an allele from F1 generation female, okay. So an allele from F1 generation A and F1 generation female gamete. Fertilization happens here. Fertilization happens. F1 means first filial generation. F1 means first filial. Okay. Now it after the fertilization, what it happens? What it happens? The 2n becomes one of the male tall, and it becomes heterozygous because this one is pure tall, this one is pure dwarf. When these two does the fertilization, one gamete from the one allele from the parent male, that is pure tall, fertilizes with the one gamete of the female, that is pure dwarf, and both becomes heterozygous tall. As the dominance character resembles in the daughter generation or the next generation, as capital T is dominant over small t, thus the 2n becomes TT. 2n is capital T small t, but their outside character, that is the by which you can understand it is tall or dwarf, it is always tall. Okay. Okay, done. Now come to the next. Now in okay. Now here stamen of male is fertilized with pistil of female. Stamen of male, that is capital T small t, with pistil of female, capital T small t. Now what happens here? This one, it is T, and here it is T. Here there is a chance of 50% segregation. 50%. Now what happens from here? The pollen grain gamete, here comes the pollen grain gamete, the pollen grain gamete, and here also okay. These two does fusion, and 2 and 3 fusion. This one is T, this one is small t, this one is capital T, this one is small t. Okay, now here T small t. Okay, now one will be capital T, another will be second and third capital T small t, another will be second and fourth small t. Okay, so what happens here if we do the chart here? Okay, now here we are doing a chart. If we draw a chart, Punnett square it is called. Okay, now here the female gamete, and here it is the male gamete, Okay, now here I am giving T, capital T, small t. Okay, now this will be homozygous tall, this is heterozygous tall, this one is heterozygous tall, this one is homozygous dwarf, Now in F2 generation there will be 3 tall is to 1 dwarf is to phenotypic ratio. This is called the phenotypic ratio. Phenotypic ratio means the ratio which determines the only the tall and the dwarf. By from the outside character you will get to see which is tall or which is dwarf. That is called the phenotypic ratio. In genotypic ratio, what it contains, what the nature of the gene. So in that case, the phenotypic ratio can also be called as the physical appearance. Okay, is also called phenotypic. Okay, now gene oblique DNA oblique genetic constituent of plant. Study is genotype. Genetic constituent क्या होता है? Is 1:2:1. What is 1? It is homozygous tall. What is 2? It is hetero tall. And what is 1 here? It is homozygous dwarf. Okay. Okay. Now come to the next question. I am giving a question. So tell me, okay, the number of phenotype. What is the number of phenotype here? Question, right? The number of phenotype. The number of phenotype here. So what is the number of phenotype? It's 2. Okay, it's 2. That is tall and dwarf. Tall, dwarf. Now question number 2, number of genotype. Number of genotype, that is T. TT, 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 3. Okay. Next question, tall. If we say tall, information is not specific. What tall? Homo tall, hetero tall. So you cannot say by simple tall. So you can say information not specific. Not specific. Okay. Whether it is homozygous tall, or heterozygous tall. Okay. Now the next question comes here. Phenotype. What is the phenotype here? Phenotype is homozygous tall and heterozygous tall. So homozygous tall and heterozygous tall. Homozygous tall, TT, heterozygous tall, TT. These are the genotype. Okay. Now to the next question, genotypic ratio and the of tall phenotypes. Genotypic, this type of question in MCQ can come. Genotypic ratio of tall phenotype in F2 generation. That is 3. How? 1:2. Why? How 1:2? One is capital T and another is heterozygous. Okay. Now note, take it as a note. 1/4 is capital T. 2/4 is capital T small t, that is heterozygous, and 1/4 is again small t. So what you get to know from here, let us do shall I need to write again? So Punnett square, I think it's not required right now because you already have got it. Okay. In the next part we come to okay. Now okay, I am drawing, because if I don't draw here you will get confused later. Okay, now I am drawing a Punnett square here of this generation. So I can write again question. 3/4 tall is phenotype. Again the question, 1/4 tall genotype. Okay. Now female gamete. So I am drawing a Punnett square. female, gamete, and here I'm drawing again capital T, small t, capital T, capital T, small t, capital T, small t, small t. Okay. So now this is homozygous tall. This is heterozygous tall. This is also heterozygous tall, and this one is homozygous tall. Okay. So in F2 generation, what we can write? 3 tall is to 1 dwarf is to phenotypic ratio. Okay. And in the physical appearance, in case of physical appearance, phenotype. Gene oblique DNA oblique genetic constituent of plant, study genotype. এক হচ্ছে one is homo tall, this is the genotype. 2 is hetero tall, and again 1 is homo dwarf. Okay, so till today I have completed for the F1 generation, that is the monohybrid cross, and in the next lecture I will continue with the dihybrid cross, test cross, back cross, all will be taught in the next lecture. So please go through the lecture, and if you find any doubt or in understanding, because it's a tough chapter in class 12, and please do not open NCERT for this chapter. If you open NCERT, you will be lost. Okay, please do not open NCERT, just understand the concept. If you do get any confusion in understanding it, please write it in the comment box. I will definitely solve it. Thank you. Good night.